And then the next presentation is then from the same group. It's Sabina and Rosa who will talk about another type of fibers and how these may alter the gut microbiota. So
Yep.
go ahead, please.
Can you hear me? Okay, so first of all, I would like to thank you, the organizing committee, for this opportunity to give this talk today here. It's a great pleasure to be here in person. So I'm Sabina. I'm a researcher in the microbial ecology and in the microbial ecology and metaomics group at NMU. The group is led by Phil Pope, that is here in the audience. And the group basically applies metaomics technologies combined with biochemical studies in order to understand how gut microbes interact with common and atypical nutrients, for instance, food additives. So we typically use, so for instance here, I'm going to show you an example on how we have combined metagenomics using both Illumina and Nanopore, as well as functional omics, for instance, transcriptomics and metaproteomics, with enzymology as well as gnotobiotic mice study to show that the inclusion of these food additives, xanthan gum, into the human diet has driven the adaptation of the human gut microbiome. So xanthan gum is a food additive that is basically an exopolysaccharide that is produced by a plant pathogenic bacterium, Xanthomonas campestris. So it's basically some kind of slime that the bacterium uses to attach to and start virulence in cabbage-like leaves. It's a heteropolysaccharide with a very complex structure. So structure consists of beta-1,4 linked glucose residues in the backbone. Then it has side chains with mannose, glucuronic acid, and mannose again, every second glucose residues. And the internal and terminal mannose can be acetylated and pyruvylated. So due to this complex structure, xanthan gum has a high viscosity, important rheological properties. So high viscosity is resistant to high pH and temperature. So it has been introduced as food additives in the human diet about 15 years ago. So it's mainly present in ice cream because it prevents, the formation of ice crystals during these cycles of freeze and thawing. Present in condiments, baked product, and is present in high amounts in gluten-free baked products because xanthan gum basically gives the fluffiness and elasticity that gluten usually provides to normal baked products. It's also present in drugs used as a carrier for controlled release drugs. For instance, is present as inactive ingredients in these medicine, e-books, and other stuff. So, is it safe? The short answer to this is that the fact that we believe that it's safe is based on papers, on studies that were conducted at the very beginning, when xanthan gum was introduced in the diet, so in 1970s or in 1990s. It's believed to be safe, that is safe in the diet, is approved by the the EFSA and the FDA, but at that point, no one basically notice an interaction with the gut microbiota. So there is only, the most recent paper is a paper from 1993 that observed some kind of bacterial adaptation. But since xanthan gum is widely present in products that are consumed in modern diets, so in Western diets, it's important to understand the effect of this food additive on the human gut microbiota. So this study started in collaboration with Professor Eric Martens at the University of Michigan when he asked his students taking the microbiology class to donate fecal sample. Samples were enriched in a minimal medium containing xanthan gum as the sole carbon source, and based on the reduced viscosity of the medium, we noticed that half of the people had a positive phenotype. So the sample was passaged 10 times to stabilize the phenotype. Then we showed that this community was able to grow on different percentage on xanthan gum. And we took samples at 4 time points for DNA and RNA extraction short-chain fatty acid quantification, and linkage analysis and rheology. So the first thing we did is to run 16S amplicon sequencing in order to understand the composition of 20 random samples. So what you see here in this bar is the relative abundance of bacteria that are present in 20 samples. But in short, what you can see is that we notice that the common factor among all these samples is this violet bar that is the a bacterium and operational taxonomic unit affiliated with the Ruminococcaceae uncultured genus UCG13. So the next step was to use DNA samples to run metagenomics. So we used the combination of Oxford nanopore and Illumina sequencing in order to obtain the complete closed genomes. And we use this as a database to map transcriptomics. So what you see here is the summary, basically, of the transcriptomic results. So we show different metabolic pathway, for instance, glycolysis, glucuronic acid metabolism, in 13 bacteria that we found in this xanthan community. Each enzyme is shown here with the enzyme commission number. The expression level is indicated by a box, and the color of the box depends on the amount of transcript that we found, so transcript per million. So in short, blue means low transcription, red means an upregulated gene. So what we see here is that all these bacteria, over time, they are down-regulating the genes, while these two bacteria, something called Bacteroides intestinalis and Ruminococcus, are upregulating the genes. We also looked into the glycoside hydrolases and polysaccharide liases, because those are, as Lars said, are the enzymes that are involved in xanthan gum degradation. And we found upregulated GH and PL in these two populations again. So these enzymes were organized in two loci, in the Ruminococcaceae and also in the Bacteroides intestinalis. They were upregulated over time. And based on the predicted function, we found out that basically all the enzymes could be compatible with the degradation of the structure of xanthan gum. We cloned, actually we ran more metagenomics on other 16 samples, and what we determined is that this Ruminococcus utilization locus is basically very well conserved among all the culture. The only thing is an insertion here of an extra gene, while only a subset of the sample contained the Bacteroides intestinalis pool, so meaning that probably this Bacteroides intestinalis is not really essential for xanthan degradation. No? So we cloned, expressed, and determined the biochemistry of all the genes. We ended up with this model in which the Ruminococcaceae is the primary degrader, using a xanthanase to degrade the polymeric xanthan. And these oligosaccharides, they can be used by this Bacteroides intestinalis that is a cross-feeder. So the oligosaccharides are uptaken and degraded intracellularly to produce acetate, lactate for the Ruminococcus, and acetate and propionate for the Bacteroides intestinalis. Then we ran a mouse experiment in which germ-free mice were colonized with a synthetic human gut microbiota containing the Ruminococcus. And then they were kept either on the xanthan diet, so the group in orange, or xanthan diet was removed, the group in blue. And after 36 days, they were gavaged with the Bacteroides intestinalis. So we determined the relative abundance of the Ruminococcus and the intestinalis. And as you can see, already in the orange diet, we observed the presence of these two populations, meaning that the xanthan is supporting the expansion of the Ruminococcus and the intestinalis in vivo, while there is no bacteria in the blue diet. So the last experiment was to use the sequence of these two loci to map reads coming from publicly available fecal metagenomic samples in order to determine the occurrence of the two loci in human populations. So the Ruminococcus, so in green you see the Ruminococcus, presence of the Ruminococcus locus that was present in Chinese, Japanese, and European and American populations, while the intestinalis in red is less frequent. But I just want to draw your attention in these three populations here. Those are agriculturalists or hunter-gatherers, so they have never been exposed to xanthagum, and indeed these two bacteria and Lerosae are not present. So in conclusion, we show that the human gut microbes do consume xanthagum, and to make a long story short, this inclusion of xanthagum in the diet may have driven this colonization and expansion of this ruminococcus into gut communities of numerous human population. So now the next question is to try to understand the effect of xanthagum on host health, because we have seen basically that 50% of the population does not have communities that degrade xanthagum. Some of them, they do not have a cross-feeder, meaning that the primary degrader could basically liberate some oligosaccharide that could interact with epithelium, gut epithelium, so maybe causing some host effect. And with this, I would like to acknowledge the memo group, so Phil and other memo members, Eric Martin's group at the University of Michigan, and other members, as well as other collaborators in this paper and the Research Council for funding. So thanks very much, and I'm happy to take the question you might have.
So, thank you very much for a very nice, interesting talk. Please come forward to the microphone if you have questions. But I could start off. It's, of course, of great interest to know more about the additives that are, well, say, generally regarded as safe. This is a common term often. So how do you have any, based on which microbes you see are able to handle this and so on, do you have any hypotheses or suspicions on where the possible health effects would be?
So these two microbes, they see, based on the metabolic output, so acetate, propionate, butyrate, they might not, directly cause a negative effect. But so we don't really know what's going on in vivo communities, because there might be other microbes in this. It's always a very complex food network. So again, the primary degrader could generate this oligosaccharide that could cause inflammation, and this is something that is not been taken into account before, because xanthagum was considered not to interact with bacteria. So it was thought to pass without any consequence for humans. So we have not done any toxicological studies, for instance, because it probably depends on how much xanthagum each person consumes. Yeah. So maybe if you eat every day a lot of this junk food, junk food, and you don't really have a gut microbiota that could help you, there might be some consequences.
Right. Some years ago, there have been several studies. There have been on like emulsifiers, and also on artificial sweeteners, and typically testing in, say, models of inflammatory bowel disease, for instance, and with the potential effects. Are you looking in that direction or elsewhere?
Yes, we are looking in that direction. So we know, for instance, emulsifier like carboxymethyl cellulose or other food additives are causing quite, dramatic negative effect on health. And we are going to use pretty much the same approach and see the interaction between this diet, microbiome, and host metabolism.
Very good. You're not very eager today, but step forward if you want to. Fredrik?
Hi, thanks for a nice talk. My name is Fredrik Miel from the Rikshospitalet. I have two questions. First,
yes.
Do you know any studies where these Rhinococcus have been linked to some kind of disease or some condition?
No, I'm not aware of any study.
No.
It's no, actually no.
No. And my second question is, because I saw on all these color charts, there were some, or I saw the Clostridium as well. It wasn't specified down to species level. But a few years ago, there was a linkage between, now I don't remember the compound, but with some compound that was found in ice cream and the increase in Clostridium. do you think, or can you speculate if there's a correlation between the introduction of xanthan gum and the...
I think, again, it depends a little bit on the community that each person has. So there might be some consequence depending on what the community is and what xanthan gum is promoting there. There might be a correlation with the Clostridium as well, but yeah, we don't really know.
Okay.
We haven't got that far yet.
Yeah, thanks.
Very good. Well, thank you very much again for, I think it's a very interesting and important field, so happy to hear more about further studies as well.